Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Molecular Microbiology. I will be doing only the biology part, and this exam was conducted in the year 2021. If you are new to my channel, please do like, share and subscribe. Then match the entries listed in group 1 with the group 2. So group 1, it is ion exchange chromatography. So it will be increasing salt concentration, that is P3. Then gel filtration chromatography is based on molecular size. Hydrophobic interaction chromatography, decreasing salt concentration, and chromatofocusing is for ampholytes. So answer is option C, that is P3, Q4, R1 and S2. Then next one, which of the following statement is false regarding disulfide bonds? So they are formed by oxidation of thiol groups. They are formed between cysteine residues. They hold the heavy and light chain of immunoglobulins together. So all the three are correct. They stabilize the T form of hemoglobin. That is not correct. So answer is option D. Then blocking the active site of an enzyme is a kind of. So it is a kind of competitive inhibition, where you block the active site of where the inhibitor blocks the active site of an enzyme. Then estrogen and testosterone are steroid hormones and most likely to bind. So they will bind to cytoplasmic receptors. Answer is option D. Which of the following is incorrectly stated regarding ABO blood group in human beings? The ABO blood group locus has 3 alleles. Yes, that is A, B and O. IA and IB alleles are dominant over small I alleles and are co-dominant with each other. So that is also correct. The person with blood type O produces both A antibodies and B antibodies. Yes. Then there are 4 genotypes possible at this locus. It is not 4 genotypes, it is 6 genotypes are possible. That is AA, AO, BB, BO, AB and OO. So answer is option D. Then a DNA molecule of 300 base pair long has 20 complete rotation. This molecule is negatively supercoiled. Answer is option A. Then next one. Dash are plasmids that are capable of freely replicating and able to integrate into bacterial chromosome. They are episomes. Answer is option B. Ashanti De Silva was the first to receive gene therapy. Answer is option A. Which of the following genes is are targeted to detect COVID-19 infection using qRT-PCR? So it is E gene, that is 1, and RDDP gene, that is 3. So answer is option A, that is 1 and 3. So coming to the next question. The core polysaccharide of the lipopolysaccharide layer of Salmonella consists of. So it consists of keto deoxyoctanoate, heptose, glucose, galactose, and acetyl glucosamine. Answer is option A. Then in gene mapping experiments using generalized transduction, bacterial genes that are co-transduced are. They are close together on the bacterial chromosome. Answer is option C. Then identify the mismatch. Staphylococcus aureus, widespread hospital pathogen. Escherichia coli, cause of urinary tract infection. Candida, opportunistic pathogen, and Bordetella pertussis, tularemia. So Bordetella pertussis, it causes whooping cough and not tularemia. So that is the mismatch and that is the answer. So option D is the answer. A vector that is specifically designed to replicate in 2 different hosts is referred to as shuttle vector. This is also a question that has been asked in other MSc entrances as well as in integrated MSc PhD entrance also of this university. Then which of the following is the only enzyme of the TCA cycle that is not located in the mitochondrial matrix? So it is succinate dehydrogenase. It is found on the inner mitochondrial membrane. So answer is option B. Which of the following statements regarding recombination frequency between the genes is incorrect? So a recombinant frequency of 0 corresponds to independent assortment. That is incorrect. Answer is option D. The next one. Among the listed amino acid, which is not an intrinsic fluorophore of protein or peptide? So it is valine. Answer is option A. Then, in a negative repressible operon, the regulator protein is synthesized as. So, in a negative repressible operon, transcription of the operon normally take place. Repressor proteins are produced by the regulatory gene, but they are unable to bind to the operator in their normal conformation. But certain molecules called as co-repressors are bound to the repressor protein. They cause a conformational change to the active site. The activated repressor then binds to the operator and prevent transcription. So the tryptophan operon that is involved in the synthesis of tryptophan is negatively controlled repressible operon. Here the tryptophan acts as the co-repressor. So the answer is option B, that is an inactive repressor. So the negative repressible operon, the regulator protein is synthesized as an inactive repressor. So answer is option B. Then which technique commonly used to study epigenetic alterations? So it is chromatin immunoprecipitation. Epigenetic alterations are age-related changes in the gene expression that harm the fundamental function of cells and increase the risk of cancer and other age-related diseases. So answer is option B. Then in proteins, N-linked oligosaccharides are attached to asparagine, ASN. This is also a repeated question. Then read the following statements and reason carefully with regard to the enzyme nitrogenase and identify the correct answer. The reduction of acetylene to ethylene is widely used for measuring the activity of nitrogen fixation. Then the enzyme nitrogenase is not entirely specific for nitrogen as the substrate. This enzyme can also reduce cyanide, acetylene and several other triply bonded compounds. So here both the statements and reason are correct and reason explains the statement. So answer is option A. The next one. The technique that uses hybridization to detect specific DNA restriction fragments in genomic DNA is restriction fragment length polymorphism. Answer is during eukaryotic replication, dash degrades the RNA primer by 5' 3' exonuclease activity. So it is FEN1. Answer is option B. Then cardiolipin is a characteristic of the membrane of, this is a repeated question, it is of mitochondria, that is 1D. Then a stretch of 25.68 kb kilo base pair is equivalent to dash mega base pair. So 1 mega base pair is 1000 kilo base pair. So 25.68 kb is 0.02568. Answer is option C. The molecule that functions as a natural thiol reductant in the cell, it is glutathione. Answer is option A. In all the examination, I have found at least one question with glutathione. Then next one. Guard cells differ from epidermal cells in having chloroplast. Answer is option B. In plants, which of the following is not an ATP hydrolyzing pump that is found in membranes? So it is aquaporins. It facilitates transport of water between cells. Then next one. Why is ATP considered the high energy molecule? Answer is the products of its hydrolysis are stabilized by resonance. Answer is option C. All these are just direct questions. Next one. Optical isomerism is shown by compounds having a symmetric carbon atom. Answer is option C. Then grapes shrink when placed in hypertonic sugar solution. The process is called exosmosis. Answer is B. Reverse transcriptase is, this is also a repeated question, RNA dependent DNA polymerase. Then nystatin is an antibiotic against fungi. Answer is option C. Then blast disease of rice is caused by a fungus. Answer is option C. Which of the following is a derivative of mycolic acid? It is code factor, because it is also a repeated question. Code factor or trehalose dimycolate is a glycolipid molecule found in the cell wall of Mycobacterium tuberculosis and similar species. Answer is B. Then the consensus sequence TAT-AAT found in most bacterial promoters approximately 10 base pair upstream of the transcription start site is called as Pribnow box. The bacterial homologue of the Tata box is called as Pribnow box. Answer is option A. Then delta G of a cellular reaction will be negative if products of the reaction have more entropy than the reactants. Answer is then, what does CT value or cutoff threshold in qRT-PCR experiments imply? CT value, it is the relative measure of the concentration of the target in the PCR reaction. So, lower the value, higher the transcript number. Answer is option A. Then acetyl-CoA, the cytoplasmic substrate for the fatty acid synthesis, is formed in mitochondria. The intermembrane space is impermeable to acetyl-CoA. Which compound is the form in which the carbon of acetyl-CoA is transported to the cytoplasm? So it is as citrate. Answer is option C. Which of the following is a common disease in groundnut? Answer is Ticca disease caused by Ascomycete, Cercospora arachidicola. Answer is option. Then, which of the following photosynthetic pigment structure is closely mimicking to the bile pigment bilirubin structure? It is phycobilins. Answer is option B. Then next one. To purify a particular recombinant protein, scientists choose to use affinity chromatography as he has nickel columns available in his laboratory. Which of the below molecule he should use to tag protein for purification using these columns? So they should be histidine tagged. Answer is option D. Then, during daytime, if carbon dioxide concentration around the leaf increases. Okay, since stomata allow the carbon dioxide to enter and oxygen to exit, therefore, if there will be a sudden increase in carbon dioxide concentration around the leaf, the stomata will get closed so that the excess gas does not enter the cell. And this will further decrease the rate of transpiration. So the answer is option D, decrease in transpiration due to closure of stomata. The next one, the enzyme used to prevent unwanted self-ligation of DNA molecules during cloning experiment is alkaline phosphatase. So it is used for the removal of single phosphate groups from the 5' end of linear vectors to prevent recircularization during cloning. Then what is Graafian follicle? So it is a mature follicle with which an ovum develops prior to ovulation. Answer is A. Then protein phosphorylation is a mechanism of regulation that is extremely important in most cellular processes It occurs through reversible post-translational modification. Answer is option B. So next question. In PCR reaction, which of the following temperature is the most suitable for annealing reaction to take place? It is 55 degrees. The others are not. Okay, so now usually it is 55 degrees. Answer is option A, 55 degrees. Okay, the enzyme specific to glyoxylate cycle is. So it is isocitrate lyase. Answer is option A. Which is the most important structure related to microbial attachment to cell? It is glycocalyx. Answer is option A. Option D. Then which enzyme catalyzes the reaction 2H2O giving 2H2O plus oxygen? That is catalase. Answer is option C. Then what would be the frequency of this particular genotype? In this particular genotype, individuals from mating of two individuals of genotype this and this. So these are the individuals that are involved in the mating. So what is the frequency of this particular genotype being obtained after mating these two? So if you cross capital A small a with capital A small a, you will get capital A small a you have to get over here. So it will be around 2/4, that is half. So half is the probability of getting capital A small a. Now if you cross small b with small b, the chance of getting small b in the final genotype is 1. Now if you cross small c capital C with small c capital C, The frequency of obtaining capital C is only 1/4. See, out of all these four genotypes, only 1/4 frequency you will get it as capital C. And if you want capital D small d crossing, you have to get capital D small d in the offspring, then it is also half probability. Now the total frequency is half into 1 into 1/4 into half, that is 1/16. So the answer here is option A, that is 1/16. Next one. Which among the following is high energy electromagnetic radiation known to arise from the radioactive decay of atomic nuclei? So it is gamma rays. Answer is option A. Then telomeres are present in eukaryotic genome at the chromosomal ends. So it is to protect them from breakdown. Answer is option B. The major role of telomeres is to cap the chromosome ends to minimize the loss of DNA during rounds of cell replication. So answer is option B. Then which of the following is a linear and polar molecule? It is N2O. Answer is option A. Then the cell organelle which show extensive polymorphism it is lysosome. Answer is C. Mycotoxins are formed during the end of the lag phase. Answer is option A. Then arginine and lysine are found in what form and behaves in what way? So they are found as positive ion and behaves in alkaline way. That is option D. Then in the context of bacterial colonization, the dash is the zone of soil adjacent to and influenced by roots. That zone is called as the rhizosphere, and where the surface of the root is known as the rhizoplane, and the dash refers to the root cortex, that is endorhizosphere refers to the root cortex. So the answer is option C, rhizosphere, rhizoplane and endorhizosphere. The next one, histones and albumins are example of simple proteins. Answer is option A. The causal organism of Kala Azar is Leishmania donovani. Answer is option C. In a redox reaction, electrons move. So electrons move from compounds having lesser positive redox potential to compounds with more positive redox potential. So answer is option B. The next one. What is plasmogamy? So cytoplasm of the two parent cell fuse together before their nuclei fuse during sexual reproduction in fungi. So answer is option B. Then cholecystokinin is an example of peptide hormone of gastrointestinal system responsible for stimulation of digestion process. This hormone is synthesized and secreted by the small intestine. Answer is option B. Hyperthyroidism is medically referred to as Graves' disease. Answer is B. Then Sabin vaccine is given to offer active immunity. Answer is B. Large seaweeds called kelps belong to brown alga. Answer is A. Through wobble, a single dash can pair with more than one dash. Through wobble, a single anticodon can pair with more than one codon. Answer is D. Which of the following hormone is derived from phenylalanine and tyrosine amino acid? So it is epinephrine. Answer is D. Then match the entries in group 1 with enzymes in group 2. So NAD plus is lactate dehydrogenase. So P3. Then selenium is glutathione peroxidase. Pyridoxal phosphate is glycogen phosphorylase and molybdenum is nitrogenase. So the answer is option C, that is P3Q1R4S2. When an aldehyde or ketone treated with alpha bromoester in the presence of zinc, it leads to the formation of beta hydroxy ester after acid hydrolysis. This type of reaction is called as Reformatsky reaction. Answer is option. Then match the type of mutation in column 1 with the major features in column 2 and choose the correct answer. So silent mutation. Silent mutation is no change in amino acid. 1, 2. Missense mutation, change in amino acid encoded. Nonsense mutation, creates translational termination codon. Frameshift mutation, shift the triplet reading of codon out of the correct phase. So the answer is option B, that is 122431143. Then which of the following genetic disorder is not related to blood? It is phenylketonuria. Answer is option C. Then a non-protein structure covalently attached to protein part of an enzyme molecule is called. It is called as the prosthetic group. Answer is option D. Then match the following for the micronutrients needed by microorganism. So chromium, no non-microbial requirement. So K5. Molybdenum is for nitrate reductase, L1. Zinc is carbonic anhydrase, M2. And manganese is for water splitting enzyme. So answer is A, that is K5, L1, M2, N3. Which of the following antibody is predominantly found in saliva? So it is IgA that is predominantly found in saliva. Answer is option A. Next one, decrease in zinc source ratio causes the rate of photosynthesis to decrease. Answer is option A. Then next one, match the following for an enterobacter grown on different media. So inosine methylene blue, EMB, it is greenish metallic sheen. So K2. Then McConkey is red to pink, L1. Salmonella shigella, it is M4, that is white or beige, and bismuth sulfite, it is mucoid colonies with silver sheen. So answer is option C, that is K2, L1, M4, N3. Then star-shaped chloroplast is seen in. It is seen in the case of Cygnema. Answer is option. Then an example of a symbiotic association of non-leguminous plant with rhizobium is. It is Parasponia. Answer is option D. Which of the following organic reaction is known as Ullmann reaction? So it is aryl iodides and bromides when heated with copper form biazyl compounds in which two benzene rings are bound together. So answer is option B. An example of point mutation. It is in the case of sickle cell anemia. Option B. Then the causing agent of citrus greening disease is. It is Candidiatus liberibacter asiaticus. Answer is option A. Then presence of which pigments is responsible for absorption of all wavelengths along the visible spectrum in red algae, giving it almost black appearance. It is phycoerythrin and chlorophyll. Answer is option D. Then gastrointestinal tract is the site of food digestion, consists of the stomach, small intestine and large intestine. The tract is rich in microbial diversity and a niche for the growth of many microorganisms. Which among the following part of the digestive system is considered as a chemostat for microbial growth? So it is the large intestine. Answer is option A. Then following are the statements on Mendel's success in formulating law of inheritance in plants. Select the combination with all correct statements from the above. So his choice of Pisum sativum for his experiment which showed well-defined contrasting character, that is correct. Then he studied 7 pair of contrasting characters individually in parents and hybrids, that is also correct. He focused his attention to many contrasting characters at a time during hybridization experiment. No, it was only to one particular contrasting character that he focused. Then he made controlled crosses, accurately maintained the records and analyzed the data statistically. So that is also correct. So 1, 2 and 4 are correct and that gives answer as option A. Then flatus is the term used for the gas produced within the intestine due to the action of fermentation and consists mainly of carbon dioxide, hydrogen and sometimes methane. Answer is option B. In pedigree analysis, if affected fathers pass on the trait on to all their daughters, then it can be inferred as X-linked dominant trait. Answer is option B. Because it is fathers pass the trait to all of their daughters. So it is X-linked dominant trait. Then identify the mismatch. Mycoplasma genitalium, smallest known cellular genome. Rhodobacter spheroides, oxygenic phototrophic bacterium. It is anaerobic phototrophic bacterium. So that is the mismatch. So 2 is a mismatch. Then Pyrococcus absci is an archaea which can grow at high temperatures, and Borrelia burgdorferi is causes Lyme diseases. So only 2 is the mismatch. So answer is option D, 2 only. Then heterocyst of cyanobacteria, it is specialized for nitrogen fixation. Answer is D. Then activation of rubisco by light is due to release of rubisco from thylakoids into the stroma. Answer is option C. Earth is thought to be dash billion years old. So it is 4.6 billion years old. The first evidence of microbial life emerges in the rock about 3.86 billion years old. So answer is option C. Then hexokinase activity in glycolysis is inhibited by glucose 6-phosphate. Answer is B. Then protein separation based on ligand specific method called as affinity chromatography, because it is based on ligand specific method. It is called as affinity chromatography. A particular protein will bind to its specific ligand. Then a binary vector. Binary vector, it is an expression vector, as you can see in the case of Agrobacterium tumefaciens vector. Then chloride that turns black on addition of NH4OH, that is HgCl2, mercury chloride. Then when phenol reacts with acid chlorides or acid anhydrides in aqueous alkali solution, it produces dash. In this reaction, the alkali first forms the phenoxide ion, which then reacts with the acid chloride. So it will produce phenyl esters. Answer is option C. Then next one. Resorcinol is very important organic compound which is used in skin related problems. How is it obtained in the laboratory? So it is obtained from metabenzene disulfonic acid. Answer is option A. Then choose the correct statement. In plants, photosystem 2 removes electrons from water and passes them to plastoquinone. The complex associated with the cleavage of water is called oxygen splitting complex, which is closely associated with pigment system 2. So statement 1 is correct, whereas statement 2 is false. So the answer is option A. And the oxygen evolving complex, also known as water splitting complex, is the portion of the photosystem 2 where photooxidation of water occurs during the light reactions of photosynthesis. It is not oxygen splitting complex, it is water splitting complex. So answer is option A, that is statement 1 is correct and statement 2 is false. Then identify the statement that is incorrect regarding genetic code. The genetic code is based on triplet of bases. Yes. The genetic code is non-overlapping. Yes. The 64 codons encode amino acid. No. It is 61 out of the 64 which represent amino acid. The remaining 3 are stop signals which trigger the end of protein synthesis, and they do not code for any particular amino acid. So option C is the incorrect one. The genetic code is unambiguous. That is also correct. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.